Good evening, gentlemen. I welcome you all for today's lecture on applications of stimuli-responsive polymers. And these to be delivered by Mr. Samath Mallamarachi, and we have his spouse Naranji. We welcome you all for today's lecture. They are all the way from USA. He is currently a PhD student attached to Texas A&M University, and he is doing research which is quite remarkable in the sense that it will be of great use in the future when it comes to practice of medicine. Now we know that when we give medicine orally, it gets into blood and goes all over the body, and if drug is required only at a certain place, right, only a small amount will go to that place, more than 99% will go to other places, right? So when you have stimuli kind of response, right, it's possible to direct the medicine to correct locations, and we can give low dosages, right? And that is of great benefit to patients, and also side effects, etc., minimized. Now he will talk about this great research area on which he is actively engaged, and introducing Samath. He was one of the most brightest students in a chemical engineering batch, where he got almost 100% average in his undergraduate work, and finally ended up with the first class. And his wife also is of the same caliber. She is also doing a PhD. She didn't start yet, no? She has started, right? So both husband and wife are doing cutting edge research, which I am sure will be of great benefit to mankind in the future. So without much ado, I would like to invite Samath to deliver his lecture. Although attendance is poor, it goes on YouTube, and the audience will be worldwide. So thanks.
And good afternoon, everyone. First of all, I would like to thank Professor Anul Pereira for inviting me to do this lecture. So today I will talk about applications of stimuli-responsive polymers. So stimuli-responsive polymers may be a somewhat new topic in Sri Lanka, but in worldwide it is widely used in mainly biomedical fields. So a stimuli-responsive polymer is a polymer which has the ability to change its physical or chemical properties under an external stimulus. So some examples for external stimuli are temperature, pH, electricity, chemical composition, light and magnetic fields. So talking about applications, as I mentioned, as Professor Pereira also mentioned, they are very widely used for biomedical applications. So among them, the most prominent application is the drug delivery, and also it can be used for enzyme activity regulation, biosensing, and also smart irrigation systems, which is somewhat outside the biomedical field. So first of all, I will talk about drug delivery, and that's the topic I will explain in most detail today. So what happens in stimuli-responsive drug delivery is that the drug is released in response to a given stimulus. So the important fact is that it can deliver precise amount of the drug to the desired location at the desired time. That means we have the control over how much drug is released and where it will be released, and also at which time and for how long the drug will be released. So that's very useful when administering a drug. So because of these features, that stimuli-responsive drug delivery has many advantages over the conventional drug delivery. One thing is avoiding drug overdose. So if I explain it further, in conventional drug delivery, a single dose of the drug is orally taken or injected to the blood at one time. So what happens is, initially the drug concentration in the body will increase rapidly, and then it will again decrease and go below the desired level. So in that case, when initially the drug concentrations goes very high, it has the ability to cause drug overdose. It frequently happens when administering insulin to diabetic patients. Sometimes because of insulin overdose, people's blood glucose level goes down rapidly, and we have heard some people dying because of that. And by using stimuli-responsive drug delivery, such kind of incidents can be avoided. And another advantage is that we can deliver the drug precisely to target location. So as we know, when someone is suffering from a disease, only a selected parts or selected tissues of the body requires medication. But if we take it orally or inject it to the blood, the most of the drug will go to different places of the body where it is not needed. That means in one hand it is a waste of the drug, and on the other hand it can also cause some side effects. By doing through stimuli-responsive drug delivery, we can control the location of the delivery. And then the other thing is maintaining steady drug concentrations over long time periods. So in stimuli-responsive drug delivery, we can control when to release the drug and how much it should be released. So we can control the stimulus so that the drug concentration is held steady for a long period of time, which is beneficial for the patient, and also reduce side effects. It is a combination of the above three factors. Since we avoid the drug overdose and we only provide the drug to the place where it is needed, we can avoid the side effects. And finally, since we are speaking about stimuli-responsive polymers, the polymers also offer the advantage of preserving the drug from environmental conditions. An example is if we take some protein drug which is taken orally, if we directly take it, it will degrade under the acidic conditions in the stomach. But if we entrapped within a polymer, the polymer can protect it from the acidic pH. So now let's look at the mechanisms of drug release. So the most widely used mechanism of releasing a drug is the expansion or contraction of a hydrogel encapsulated in the drug. So in this case, the drug molecules are entrapped within a porous hydrogel matrix. And when we apply a stimulus, depending on the type of polymer, the hydrogel can expand or contract. So if it expands, the pore size will become larger, and then it becomes easier for the entrapped drug molecules to diffuse out of the gel. And if it contracts, the water will come out, and the drug also will come out along with it because of the squeezing of the gel. So both of those, both expansion or contraction has been successfully used for drug delivery. And the other thing is opening a polymeric membrane surrounding the drug. So in this case, we use a membrane instead of a hydrogel, and the reservoir containing the drug is covered by this polymeric membrane. And this membrane acts as a gate which responds to a stimulus. So when we apply a stimulus, the gate will, the membrane will open and allow the drug to diffuse out of the reservoir. And when sufficient amount of drug is released, we can remove the stimulus and then close the membrane. And finally, there is the erosion of the drug embedded hydrogel. In this case, drug is again encapsulated in hydrogel, and this hydrogel is designed so that it will erode when it reaches the desired location or desired conditions. An example is that when treating diabetics, there are some hydrogels which erode under hydrogen peroxide. So in this technique, this hydrogel is embedded with insulin and glucose oxidase, and when the glucose level gets high, the glucose oxidase will convert glucose into gluconic acid, and again, or as a side product, hydrogen peroxide will be produced. And this hydrogen peroxide will erode the hydrogel containing insulin, and it will release the insulin. However, compared to the first two mechanisms, the disadvantage of this erosion based hydrogels is that it is irreversible. That means once it gets eroded, we cannot use it again. So when using hydrogels for drug delivery, most preferred methods are expansion or contraction of a hydrogel or using it as a gated membrane. So now let's look at several applications of stimuli-responsive drug delivery. So the most common application is the responsive insulin delivery using hydrogel entrapping insulin and glucose oxidase. So since this is the most common application, I will describe it in more detail in the next few slides. And then, as I told before, we can encapsulate protein drugs in pH responsive chitosan hydrogels for intestinal drug release. So in this case, what happens is these hydrogels are stable in a low pH. So in the stomach they will not degrade, and thus the protein will be protected from the acidic conditions of the stomach. However, once it goes through intestines, the hydrogel will dissolve at the comparatively higher pH, and the protein drug can be released. So in that case, we can protect the protein drug from the acidic conditions in the stomach. And people have also achieved brain targeted delivery of anti-epileptic drugs by entrapment within angiopep conjugated electroresponsive hydrogels. So in this case, angiopep acts as the targeting molecule to target brain cells, and electrical stimulus acts as the trigger of the trigger to release the drug. And people have also used stimuli-responsive polymers to treat burn wounds. Mainly thermoresponsive polymers have been used for that. What happens there is there are some thermoresponsive polymers which become liquid at high temperatures. So as we know, a burn wound is at higher temperature than the other parts of the body. So when we infuse this gel with the medication and apply it to the burn wound, it will become liquid and flow all over the infected burned areas, and thus all the areas which are burned can get infused with the drug. So these are some of the wide applications of stimuli-responsive drug delivery, and there are also some like this has been done, applied for many other applications also. So now I will explain what happens in stimuli-responsive insulin delivery. So first, let's say the blood glucose level is increased. So as I mentioned before, this system contains of the porous hydrogel, pH responsive hydrogel, glucose oxidase enzyme, and insulin. So when the glucose level gets high, the excess glucose will be converted into gluconic acid by the glucose oxidase enzyme. So as the name implies, gluconic acid is acidic, so it will reduce the pH of the blood, and this resulting drop in pH will cause the deformation of the hydrogel, because the hydrogel is pH responsive. So as a result, when the hydrogel is deformed, the entrapped insulin will be released. So now let's say the insulin is released. So once the desired amount of insulin is released, the natural metabolism of the body will reduce the blood glucose levels, and once the blood glucose level reaches the acceptable value, the enzymatic reaction will stop, and then it will the hydrogel also reach converted into the original state, and the release of enzyme will be insulin will be stopped. So in this case, in like this way, we can use it over multiple cycles. So this is the graphical representation of what happens there. So initially glucose, both enzyme and the insulin is within the hydrogel matrix, and then glucose will diffuse and react with the enzyme, and as a result, the result of the reduction of pH, this will swell, and insulin will move out of the gel. And this is some experimental data which has been obtained from literature. So people have shown that at high glucose concentrations, high amount of insulin is released. That means we can control the insulin released depending on the blood glucose concentration, and this has been tested both in vitro and in vivo. So when speaking about stimuli-responsive drug delivery, an important parameter is the reversibility. So reversibility means we should be able to stop the release of the drug at the necessary moment and again restart the drug release when the stimulus is applied. We should be able to reuse it over multiple cycles. So this is an example of a reversible system. So you can see that the electrical stimulus is applied on a stepwise method, pulsatile method. So when the stimulus is present, we can see that the protein is released at a high rate, and when the stimulus is not present, the protein release is almost negligible. So this kind of behavior should be present for an effective stimuli-responsive drug release system. And there are some drawbacks of stimuli-responsive drug delivery. The main drawback is the slower response time. Why I say this is, it takes some time for the polymer or hydrogel to deform under stimulus and release the entrapped drug. So that response time may be too late when treating a critical patient in a critical condition. And then there are concerns about biocompatibility. So as I know, most of the polymers used for these systems are non-toxic, but when using them in vivo conditions, there may be other issues. So extensive studies has to be done to make sure that they can be used in vivo. So now let's move on to the second application, which is using stimuli-responsive polymers for enzyme activity regulation. So here what will happen is, the stimuli-responsive polymers will act as reversible switches of enzyme activity. So what happens here is that the access of the substrate to the active site is manipulated, and as a result the reaction rate will be controlled. So this has been successfully tested using thermo and photoresponsive polymers. So I will give an example using a thermoresponsive enzymatic switch. So this switch has been done using a thermoresponsive polymer called poly-N isopropyl acrylamide. This polymer has the ability to rapidly shrink beyond a temperature called low critical solution temperature. So let's see what happens here. On the left side of the figure, we see below the low LCST. So when it is below the LCST, the polymer is in expanded state. So even though the polymer is attached to the enzyme, the active site is unblocked. So as a result, the substrate can attach to the active site and the reaction can occur. However, when we increase the temperature to some value above the LCST, the polymer will collapse, and the collapsed polymer will block the active site, and as a result the reaction will stop. So and this can be used done vice versa also. So again, if we reduce it below LCST, again the polymer will expand and the active site will be uncovered and the reaction can happen. So this in this way it can be used to switch on and switch off enzymatic activity over multiple times. So these enzymatic switches is very new development. So they haven't been in much use in the large scale applications. However, there are several potential applications for this. One thing is the regulation of the activity of alkylase enzyme used to degrade cell walls during protein extraction. So let me, if I explain this further, when extracting intracellular products or products which are produced within the cells, we have to first degrade the cell walls to take out the intracellular product. So for degrading cell walls, we can use different methods such as homogenization, bead milling, and one such method is the enzymatic degradation. And alkylase is one enzyme which can be used for that. However, the problem with this alkylase enzyme is that when after it degrades the cell walls, the alkylase enzyme will start degrading the proteins inside the cell. This is undesirable because in most cases the product we desire is one of the proteins inside the cell. And by using this stimuli-responsive polymer, what we can do is we can entrap the enzyme within the stimuli-responsive polymer, and as soon as it breaks the cell wall, we can stop the reaction. And thus preserve the proteins inside the cell. The next thing is precise modulation of the reaction. The proteins need to be degraded to peptides of specified size. So as you know, if we directly degrade proteins all the way, it will yield amino acids. But there can be there are some occasions where we have to get peptides of a specific length. So in that case also we can use these enzyme switches to stop the reaction at the desired value. And then again we have the on-off switching off. We can use it to switch on or switch off any industrial enzymatic reaction without denaturing the enzyme. And third application we are talking about stimuli-responsive polymers is the biosensing. So in biosensors, the stimuli-responsive polymers, or rather specifically we use the electro-responsive polymers, will be used to immobilize the receptor species. So in most biosensors the receptor is an enzyme, but there are some occasions where we use other molecules or cells as the receptors. So we use the electro-responsive polymers to attach the receptor to the electrodes. The advantage of this is it facilitates good electron transfer between receptor and the electrode. So if I explain it further, in order for the sensor to work, the electrons produced at the receptor needs to be transferred to the electrode. So that doesn't happen without any connection. In this case, the polymer can be used to act as a conductor between the receptor and the electrode. And if there are other techniques also which can be used, such as molecular wiring, and our polymer also can be used as to give conductivity. And the other advantage is that it can protect receptor species from environmental conditions. So when measuring some measuring blood glucose or any other biological molecule, the sensor may be subjected to some conditions which may lead to degradation of the receptor species. So by entrapping it immobilizing it within the electro-responsive polymers, the receptor species also will be protected. And talking about some other biomedical applications, people have tested the stimuli-responsive polymer-based stents which can expand at the desired location by application of the stimulus. And also people have used SMP-based clot extraction devices. So what happens here is given in this figure. So initially the clot extraction device is in the rod form. However, when it comes to the blood clot, a thermal stimulus is provided, and at the application of a stimulus, the device will obtain a screw-shaped form. So with this screw-shaped form, it can pull out the blood clot from the blood veins. So these are the type of applications of stimuli-responsive polymers in biomedical fields. So and they can be used in agriculture also. So what we do is somewhat similar to drug delivery. The only difference is that instead of the drug, stimuli-responsive polymers are used to for the controlled release of fertilizer and water to the crops. So using that also we can make sure that only the desired amount of water and fertilizer is applied to the crops, and they are applied at the desired time. So these are the references I have used, and I would be glad to answer any questions you have regarding this presentation.
At the moment, do they have this kind of drug delivery system in operation at the moment? Is it being used anywhere?
The polymer-based drug delivery systems, I don't know whether they are available in the market, but people have done many research, mainly regarding the insulin delivery. So people have tested insulin patches where it can externally include, and also injectable microgels where it will microgels will release the insulin at the desired location. However, I do not know whether they are available in market yet.
So, what is the research you are doing? What kind of is it insulin delivery or?
So initially I was working on insulin delivery, but then I shifted towards regulation of enzymatic activity. I'm using the same kind of polymer, different applications, because insulin delivery is somewhat now becoming well explored area.
So there is this insulin in the market.
Yeah.
There is 70-30.
Yeah.
There is 70 will get released within so many hours. Then the balance 30 will get released after so many hours.
Yeah.
So is it something like this?
No, like 70-30 means so in that case it does not depend on the blood glucose level of the patient. So here
It's timing.
Yeah, it's timing.
Right.
So what we do here is, so if we take a patient with blood glucose level of 400, he will need a high amount of glucose, high amount of insulin very rapidly. But a patient with blood glucose level is 200, a smaller dosage is required. So by using stimuli-responsive drug delivery, we can achieve that. So one such figure is included here. So at 400 milligrams per liter, it releases a very high amount of insulin. So that which is required for such patients. Sorry. So the method of delivery, it can be used in different methods. So people have done different research. One thing is the injectable microgel. So a very small amount of gel which can be injected to the blood, and it will go to the pancreatic cells and can release the drug, release the insulin. And another method is the through the skin. It will, in that case, we have to attach some molecules to give skin permeability through the skin, and again when a stimulus is applied externally, it can release insulin. Yes, yeah. Sorry. Yeah, if you use a patch, it will be in the bloodstream. So if we can for targeting also, we can add some molecules. So stimuli-responsive drug delivery means like it has a wide area. So for targeting, we can in some cases we can use targeting, but in some automatically it will target. As I said, in drugs which are released in intestines, the aim is to target the location, but mainly in insulin delivery, the main target is to release the required amount of insulin. So however, we can use target target the location also, but for that we may need to use another attach another molecule or something like that.
Insulin also is required by the whole body.
Yes, yeah,
No
in
need
that
to
case,
target.
yeah,
All the cells should get it.
yeah.
Now, for example, there are very these intestinal diseases.
Yeah.
Where you have, for example, peptic ulcers, and
Yeah.
also you have various, various growths
Yeah.
and things like that. Now, can you all have that? I mean, develop this system to target that kind of thing. Otherwise, it's a major surgery and lot of.
Yeah, so if there are medications which can be taken orally, we can use these stimuli-responsive polymers to release them exactly at the intestine.
That location.
Yeah.
So how can you identify the location? I mean, should it be some kind of, is it enzymatic or acidic or?
So we have to decide the. So we have to first identify the parameter, the conditions at the.
Special parameter.
It is maybe pH or presence of some specific chemical compound, and once we identify that parameter, we can design the polymer to undergo change under that. Yeah. Sorry.
It's encapsulated the capsules.
Yeah.
What is the size of the smallest?
So people have used microgels. That means they can be directly injected to the blood. So they are very small microbeads.
Are they of molecular size or?
No, not molecular size, but yeah, small. Not nano scale. So people haven't still done much work on nanoscale polymers, but people have used nanoparticles, which are not polymers. But the polymers mainly they have used the micro scale.
And these polymers are flexible so that they don't get stuck anywhere. They can go through the heart and all over the place.
Yeah, usually like if it is a microgel, they can pass.
There's no
by,
rigidity.
yeah. Huh?
There's no rigidity.
No, it's like
blood it flows.
yeah. So that has to be tested for each application. So I have given only a general overview. So I haven't found any of these experimental done on humans. So people have done it either on like in vitro, that means like outside the living body, or within like animals like rats. So it may take some time to implement this on humans.
So you all are at the moment studying drug application on rats or guinea pigs or?
No, I'm doing it in vitro. Yeah.
So that is you are looking at say in vitro, maybe on the pH and which is
Yeah,
stimuli,
yes.
how it's released.
Yes.
And then you can finally apply it to an
Into
animal.
the-
And
yeah.
Now this polyacrylic acid which you said,
Yeah.
I mean, is it a proven non-allergic polymer? I mean, they've been used.
So it has been used on rats and those animals without
So the polymer
significant.
without any drugs that been injected into.
No, with the drugs. So they have
no
also-
allergy.
No allergies.
no.
But there have been some instance of irritation and stuff. But yeah, still for use in humans, it has to be done. Undergo.
It doesn't get digested.
No.
It just remains inert throughout.
Yes.
There is no chemical reaction between
No,
the
it's
gel and the
yeah.
environment.
No, it's already formed polymers and it's very
It's a stable polymer.
Yeah, stable polymer.
It says nanocapsules.
Sorry. Yeah. No, it's the enzyme nanocapsules. The enzyme is in the nanoscale, in the form of nanocapsules, but the polymer is in the as microgels. No, that means that they have, for extra protection, they have entrapped the enzyme within nanocapsules before adding it to the polymer.
So actually the polymer is outside,
Yeah.
the medicine is inside.
Yes. Yeah.
And it's not a rigid thing, no, it's flexible.
Yeah, flexible. It can. It's a kind of gel-like thing. Yeah.
Actually, there was a presentation. Now, cancer,
Yeah.
chemotherapy.
Yeah.
Now, cancer cells are located in various places, no?
Yeah.
Right? And the target, I mean, what we should do is the drug should be delivered only in the vicinity of the cancer cells.
Yes.
So that the cancer cell can be destroyed without destroying the normal cell.
Yeah.
Right?
Yeah.
So, is it, possible, that kind of thing?
It is possible, but first we have to identify like something which distinguishes the cancer cells from the
rest.
yeah. If the cancer, if we can find a specific chemical compound which is found only in cancer cells, we can design a polymer to respond to that. Yeah.
So that is the first thing.
Yeah.
But generally what they say is cancer cell absorbs nutrition at a higher rate than the rest.
Yeah.
So when you when you introduce a toxic material in the blood,
Yeah.
when it goes all over the body, but other cells don't consume it much.
Yeah.
But cancer cell consumes it more.
Yeah.
Because it has a kind of what do you call parasitic effect.
Yeah.
It tries to absorb all nutrition. Right?
Yeah.
And then due to taking a higher dosage of poison, it dies.
Yeah.
The other cells don't consume so much, so that they don't die.
Yeah. I think this kind of method techniques can be used, but yeah, we have to study about that.
But then they don't need to have special thing.
Yeah, but still, yeah.
Will eat much more than the rest.
Yeah, but yeah, even though the normal cells also get destroyed when taking chemotherapy, so
But then
Yeah.
when you give chemotherapy, patients lose their hair, everything, and then finally after the cells are dead,
yeah,
then they are rejuvenated. They start,
Yeah.
the hair grows like when they were young.
yeah.
So what are the kind of futuristic situation? How long will this system take to be practically implemented on human beings?
So since the drug delivery systems, they have been tested on rats and the lab animals, so I believe the next step will be
So
implementing in
how long does it take?
I think my guess is that within the next 10 years, those systems will be available in the market. Yeah.
Because they have the rapid approval
Yeah.
system,
Yeah.
terminal patients or patients
Yeah.
who are about to die,
Yeah.
they don't mind being tested.
Yeah.
So they say, okay, you can test on us. And
Yeah.
even if we die, it will be give information for all to develop it for future generations.
Yeah. So already there are glucose responsive insulin pumps. So they get the signal from the glucose sensor and it can pump the required amount. But the pump has even slower response time than the polymer. So the polymer will be somewhat an improvement from the existing pumps. The drug delivery. So stimuli-responsive drug delivery, people have started doing it around 2000. So people have now they have developed it to the very advanced systems, but using it for controlling enzymatic activity is somewhat new. It has been only been there for about like 4 or 5 years.
So what you are saying is even enzymatic reactions inside the biochemical reactions happening inside the body.
Yeah.
If there is abnormality, you can control it by this kind of thing.
Yeah. So at this moment it will take quite a lot of time to implement it within the body, because so far all the works has been done on like outside the mainly on in industrial processes. So applications like that, in vitro applications.
So in vitro application, how are you simulating the conditions inside the animal or human anatomy?
When insulin delivery, so we cannot exactly simulate the exact conditions, but what we do is we consider the important chemical parameters. So when considering insulin delivery, we look at the pH and the glucose concentration, the presence of other inhibitors, so insulin inhibitors, so stuff like that.
So I all doing research in affiliation with pharmaceutical companies?
No, it's
University research.
yeah.
So Texas A&M, is it a high class university? This kind of your university is
Yeah, it's a somewhat high-ranking university.
Texas A&M.
Yes, A&M, yeah.
Recently there was another lecturer who came.
Ah.
I forget his name.
On like engineering.
Mechanical.
Varuna Kulatira.
Yeah.
He's in mechanical. Yeah. Yeah, it does many good research on the biomedical fields.
The other presentation I listened to some time back. They were talking about nano.
Yeah.
And they were saying using nano iron particles, they can put the drug into, attach the drug into nano iron particles, and then it will go all over. And if they want to pinpoint to one place, they introduce a magnetic field
Yeah.
at that location with an external device,
Yeah.
right?
Yeah.
And then all the particles will get accumulated there.
Yeah, that's also stimuli responsive. Here we use polymers, but people have done
Even
using
polymer you can mix up
Yeah.
iron so that there is magnetism.
Yes. So people have used nanoparticles also.
Then
The iron particles.
will be secreted out.
No.
It will go out with excrete. Only heavy metals will get stuck in the body. Iron is a small. So I think cancer, your area is very important area would be cancer.
Yeah. So in my lab, my supervisor is mainly looking at the fundamental level. He's not doing. Our lab does not have the facilities to do it on animals.
You all have a medical faculty at least.
We have a medical faculty, but here.
You can collaborate.
Yeah, we have the chance to collaborate. Yes. Yeah, and so I would like to give some introduction about my lab also. So so I work under a Sri Lankan professor called Dr. Sandun Fernando. He graduated from Peradeniya University from agriculture engineering department, but he's now mainly working on the chemical and biomedical kind of fields. So now I'm working on using stimuli-responsive polymers for drug delivery and regulation of enzymatic activity. But in addition, in our lab we do research on fuel cells, mainly biological fuel cells, and then separation of proteins and biosensors, as I mentioned, the glucose sensors, those kind of stuff mainly.
Now, is it possible for these your biosensors to have a kind of communication with the central and peripheral nervous system of a human being, so that the stimulus, the pulse, the signal can come from the brain itself? Identifying that there is a disturbance.
Yeah. I haven't found anything.
Are there research going on in that direction?
That's somewhat outside my general research area, so I haven't looked much into
My brother is a neurosurgeon, so if you want, he is also very interested.
Right.
He actually started the collaboration with University of Molecular Electronics
Yeah.
to develop biomedical stuff like say oxygen sensors and
Yeah.
these things.
Yes.
But here the progress is a bit slow.
Yes. So I have some friends in the electronic department. They developed a system which could identify brain signals of a patient who is paralyzed. So they could detect the brain signals and identify what the patient wants, because the patient cannot talk or do anything. Yeah, they can get a rough idea. Yeah.
Stephen Hawking's.
Yeah.
So Stephen, you all haven't studied that kind of thing.
No, it's
Stephen Hawking's 100%
yeah.
paralyzed.
Yeah.
even till death he was communicating with
Yes,
a
yeah.
special brain signal,
Signal, yeah.
analyze and some device.
Yeah, mainly they are looking at the signals in the brain and also like where the person is looking at. So those by analyzing those two, they are able to detect what he is trying to communicate. But yeah, that's more of like kind of electronics side research.
Any more questions?
It looks like there are no more questions. Shall I conclude
Yeah.
this? Yeah. And again, thank you very much, Professor Arnold Pereira, for inviting me to this lecture. It's really great.
Thank you very much, Samarth, for our presentation, which was very interesting and futuristic as well. So what I can say is that these chemical engineers are people with knowledge in various directions, kind of wide scope, so they can get into various fields of research and even get involved in a lot of various industries. Now, example, Glaxo, we have about 12 chemical engineers, and chemical engineers are the people who are manufacturing drugs, because it's a chemical engineering operation. So unfortunately in Sri Lanka we have chemical engineering has not progressed much, because everybody once they get out of the university they confine to their own private activity. They never come forward and come out with the ideas and try to develop the industry and so on, right? So we at the Institution of Engineers Chemical Engineering section, we try to invite whenever an alumni or some chemical engineer or even probably some other engineer or expert comes, we organize lectures by the sectional committee, so that the knowledge can be disseminated and our local researchers also get new ideas of how to do new research and do something with the knowledge they have gathered by their education. So in that sense we are very thankful to Samarth for his field, I mean his research, which is I think quite innovative and maybe will be very very useful in the future for drug delivery in human beings, animals, plants and various things. And one thing I should say is that he was, I could mention about his achievements earlier, he was a highly talented undergraduate. He got involved in all our undergraduate activities, research, etc. and he won several awards, Olympiad. Yeah, he was also very good orator and so on, right? And his wife is also, we are very fortunate to have her here, was very very active, and she was one of the main organizers of all those student activities and so on. And now they are doing well in USA. Texas A&M University is one of the best universities in engineering and many fields in the world. So I hope you will be able to do much, very much more during your stay in USA, and maybe we hope you will be able to come back and serve the nation, right? But serving the nation you can do from anywhere, right? Now it's a global village, right? Wherever you are, even from Antarctic, you can serve Sri Lanka, right? So once again, thank you very much for your presentation, and we should thank all of you all, right? Despite your busy schedules, you have come here and you have participated in this seminar, which is a very very kind of encouraging thing, because we should have participants when there is a lecture, so that the lecturer also becomes motivated to lecture, and also the field of engineering will advance because more information is disseminated, and that will, it acts as an impetus to propagate engineering knowledge, right, and also get people motivated in the field. So we will have another lecture very soon by our engineer Mahavat and Forbes and Marshall's team, energy engineering, right, where they will talk of how energy can be produced at low cost, how thermal and electrical energy can be produced at low cost, what are the fuels to be used, right, and what are the problems associated with in the industry, how the industry is at the moment energy situation in the country, and how industries are adversely affected, right. If the energy cost is high, industries go down, right. So we will have a lecture in end of July or early August, right, and we have our chairman of the chemical engineering section committee, engineer Neil Vikramasinghe, technical services manager refinery, who is the main our boss here, right, and he is the chairman of the section, and I would like to invite Neil to hand over present the token of appreciation to Samarth in recognition of his presentation, and we hope all your future research and other activities will be of great success, and you'll be able to serve the entire world, right. Thank you.